Afternoon, everyone. My name is Allison Clune, and I'd like to welcome you to the Indoor Air Quality Science webinar series hosted by US EPA's Indoor Environments Division. Today's webinar on the formation of secondary byproducts from a commercial oxidant-generating electronic air cleaner will be presented by Dr. Sally Ng. Dr. Ng is a professor and Tanner Faculty Fellow in the School of Chemical and Biomolecular Engineering and the School of Earth and Atmospheric Sciences at the Georgia Institute of Technology. She earned her doctorate in chemical engineering from the California Institute of Technology and was a postdoctoral scientist at Aerodyne Research Incorporated. Dr. Ng's research focuses on the understanding of chemical mechanisms of aerosol formation and composition, as well as their health effects. Her group combines laboratory chamber studies and ambient field measurements to study aerosols using advanced mass spectrometry techniques. Dr. Ng serves as co-editor of Atmospheric Chemistry and Physics and a member of the editorial board of Scientific Reports and ACS Earth and Space Chemistry. Dr. Ng's research contribution has also been recognized by the Sheldon K. Friedlander Award and the Kenneth T. Whitby Award from the American Association for Aerosol Research, the EPA Early Career Award, the Health Effects Institute Walter A. Rosenblith New Investigator Award, and the NSF Career Award. Thank you so much, Sally, for being here with us today. Before we get started, I would like to remind everyone that this webinar is being recorded and will be made available on the EPA website at a later date. I'll also take this opportunity to mention that you can find more information about today's topic, air cleaners, HVAC filters, and coronavirus on the EPA coronavirus website. And we'll put that link in the chat box for you. Please note that today's presentation reflects the professional opinion of the speaker and does not necessarily represent the position or policy of the US EPA. If you have a question during today's presentation, you can type it in the Q&A pane on the right-hand side of your screen at any time. Questions will be read to the speaker for discussion at the end of the presentation. Given the size of the audience, we may need to combine some of the questions in the interest of time. If you need any technical assistance, you can also submit those questions through the Q&A box. We look forward to hearing from you. So, Sally, thank you again for speaking today. And without further ado, I'll hand it over to you to begin the presentation.
Thank you, Allison, for the nice introduction and the opportunity to talk about some of the work that we have been doing recently. So today I'm going to talk to you about the formation of secondary byproducts from a commercial oxidant-generating electronic air cleaner. So first of all, just a little bit of background introduction. So today we're going to talk about a lot about aerosols and particulate matter. So particulate matter are these tiny particles suspended in the air, and they're everywhere around us. There are many different sources of particulate matter, such as from car exhaust that you can see here, from sea spray aerosols, from wildfires, from different emissions from biogenic sources, volcanic eruptions, dust, and even from your everyday cooking activities. And in this talk, we are using particulate matter and aerosols interchangeably. So sometimes we refer to particulate matter or aerosols, but they are technically the same thing. So in terms of particulate matter or aerosol health effects, so EPA regulates outdoor PM 2.5 and PM 10 concentrations. So PM 2.5 corresponds to particles with 2.5 microns smaller in diameter, and PM 10 corresponds to particles that are smaller than 10 micron in diameter. So just to give you some sort of reference, our hair is about 50 to 70 micron in diameter. So these are really tiny little particles that are suspended in the air. So particulate matter contributes to a lot of the health problems that we are all experiencing today. About 9 million of deaths worldwide can actually be attributed to ambient PM 2.5 pollution. So if you look at this bar chart here, which is from the global burden of disease, you can see that air pollution ranked number 5th in the global burden of disease, showing that it is contributing to a lot of mortality and morbidity that we are experiencing in the world today. So a lot of the particles we are breathing actually are not emitted directly, but they are generated from a series of chemistry in the atmosphere. So in terms of submicron particles, which are the particles smaller than 1 micron in diameter, the cartoon here illustrates several different important types. So you see the red is sulfate, nitrate in blue, ammonium in orange, and chloride in purple. So these are inorganic aerosols. And they are formed from the oxidational reaction of different gases in the atmosphere, such as sulfur dioxide coming from coal-fired power plants can be oxidized to form sulfate. And nitrogen oxides coming off from car exhaust can be oxidized to form nitrate aerosol, and so on and so forth. So these are all inorganic aerosols. But there's another class of aerosols called the organic aerosols. And when I say organic aerosols, we are not just referring to one compound. It is actually made of hundreds and thousands of different compounds. And a lot of them are coming from the oxidation of VOCs in the atmosphere, which are the volatile organic compounds that are emitted by different sources. So we want to talk a little bit more about these atmospheric organic aerosols because they're such an important class of aerosols that we are breathing in every day, no matter where we are. So in this cartoon here, you will see the different sources at the bottom, in which you see you got the trees, you got the wildfires, the cars, and industries. So many of these sources can emit particles directly. So if a particle is emitted as a particle, we call it primary organic aerosol. So it is just emitted as a particle itself. So these are represented by the gray arrows that you're seeing here. But many of these sources can also emit gas compounds, like the volatile organic compounds, the VOCs. So if it is from trees, we call these the biogenic VOCs. If it is the anthropogenic VOCs can be emitted from car exhaust or in factories. And because the atmosphere is so oxidizing, once these VOCs are emitted in the atmosphere, they can be oxidized by these three main oxidants in the atmosphere in red. The first one is the hydroxyl radical OH, we'll talk a lot about today. The second one is ozone. The third one is the nitrate radical NO3. So these oxidants are going to react with your VOCs. And once these reactions happen, you're going to form many different classes of compounds. So if a compound is really volatile, you'll remain in the gas phase. So you just hang out in the gas phase. If it is semi-volatile or it is really low volatility, then instead of being a gas, it actually wants to be a particle. So how do you become a particle when you are a gas? So there are two different ways. The first way is that you go through this process called nucleation. So you're going to make new particles by yourself and just generate new particles. Or if you see another particle close to you, then you can actually condense on that particle. And this is kind of the process that we call it as the gas particle partitioning and forming secondary organic aerosols. And it is secondary not because it is not important. It is secondary because, as you see in this schematic, it is formed from a series of complex chemistry in the atmosphere. So if we look at everywhere around the world today, the organic aerosols are everywhere. So you see a series of pie charts in this map, and these pie charts correspond to the composition of the submicron aerosols, non-refractory aerosols that we are breathing today. And the green part of the pie are the organic aerosols. And you can see that organic aerosols are really everywhere, and they account for about 50% on average of the submicron non-refractory particle mass that is everywhere around the world. So that's so much about the outdoor aerosols. What about in an indoor environment? In an indoor environment, again, there are many different types of aerosol sources. So they can be directly emitted from different activities such as cooking, cleaning, as you can see from this schematic here, or you can get particles from resuspension of dust from carpets or floors and other surfaces in the indoor environment. And if you have windows open or you have the leaks in the house or the buildings, you can also get the incursion of ambient air, so the outdoor aerosol can also come in. And as we just saw in the previous introduction slides, the same thing can also happen in the indoor environment, in which you can have aerosols that are formed from the oxidation of these volatile organic compounds, VOCs, followed by these gas particle partitioning process that I just discussed. And of course, during the course of the pandemic, the aerosols coming from us humans that we emitted during our breathing, talking, singing, and everything else caught a lot of attention because of the transmission of the disease. So the chemistry in an indoor environment is also very rich, just like the outdoor atmosphere. So there can be many chemical and physical transformations that can happen in an indoor environment. And at the same time, we can also have the same type of oxidants that we show here as the outdoor environment. Again, we got the hydroxyl radical, we got the ozone, we got the nitrate radical as these major three oxidants. And there are many different reactive species and complex chemistry that can take place in the indoor environment, including the VOC oxidation, photochemistry, and surface chemistry. So of course, during the COVID-19 pandemic, there has been increasing recognition of the importance of the airborne transmission of the disease. And the particular aerosols emitted by human beings was considered as the major route of transmission of the disease from just breathing, talking, and singing and other activities. So this also resulted in the huge increase in demand for different air cleaning technologies. So technically speaking, what we're trying to do is that you want to remove the indoor pollutants, either it's the VOCs, the volatile organic compounds, or the particles, or we also want to inactivate any virus and pathogens and so on. So broadly speaking, there are several different technologies that have been used to remove particles and gases pollutants. So for the particles, there are two main types. You can use mechanical filtration or use just like using a HEPA filter or MERT filter. And you can also use electronic air cleaners such as ionizers or electrostatic precipitators. And for the gas pollutants, again, you can also make use of absorbent media air filters such as the use of an activated carbon. And there are also different electronic air cleaning devices that generate ions, reactive species, or other chemical products, including the photocatalytic oxidation, and ozone generator, hydroxyl generator, and so on and so forth. And some of these devices have been shown to be capable of inactivating virus, bacteria, and other bioaerosols. Although the efficacy in the real environments have been debated because their effectiveness appears to be varying with operating conditions. And as of now, there are no standardized testing conditions for all these different types of cleaners. And if you missed it, recently there's also a very nice EPA IAQ webinar on air cleaning by Professors Elliot Gaw and Brent Stevens, and I put the link here, and they spend a lot of overview in terms of navigating the landscapes of air cleaning technology. So if you missed it, you can check it out using this link. So there have been several studies in terms of reporting secondary product formation from electronic air cleaners. And I list here some relevant studies in terms of these formation from ionizers, PCOs with UV lamps, electrostatic precipitators, and plasma systems. And these two figures are coming from two publications. So let's take a look at this one on the left here. This paper reported the use of an induct bipolar ionization device. And the y-axis, you can see here these bars representing different types of VOCs and oxidized VOCs. And the one on the left here shows the upstream of this bipolar ionization device. So this is the composition, and this is the downstream of the device. So you can see that the composition of these VOCs and oxidized VOCs looks quite different. The abundance looks quite different, suggesting that there is some chemistry happening in the use of these bipolar ionization device. And the one on the right, it is also another study that looked at the impact of an ion generator. And you can see that they did the experiments in 4 different rooms. So you got 4 different set of bars. And the left bar is without this ion generator, and the right bar is with the ion generator. And the researchers observed increases in the particle concentration, ozone concentration, and also some of these OVOCs, oxidized VOCs, such as formaldehyde and nonenal. So these studies really indicate that there's some sort of chemistry that is happening from the use of these electronic air cleaners. And very recently, just last month, Doug Collins and Delphine Farmer published a very nice perspective article in Environmental Science and Technology calling to these unintended consequences of air cleaning chemistry, and really highlighting the needs to have more studies, like peer review studies, to kind of really understand all this different chemistry that can be induced by different cleaning technologies before they are widely adopted and used in our everyday life. So in this particular study, we're interested in evaluating the effects of a commercial electronic air cleaner inside an office. And the specific electronic air cleaner that we tested is a hydroxyl radical generator, OH. So OH is hydroxyl radical. And this device generates OH, hydroxyl radical, and hydrogen peroxide, H2O2, via photocatalytic reaction. So our goal is to measure, if any, gases, particles, byproducts using the suite of analytical instruments that we have in our lab, including a large number of advanced mass spectrometers. The question to answer is there any indoor chemistry that is induced by the operation of this air cleaner and potentially leading to the formation of different byproducts? So here is the experimental schematic setup. So we put this electronic air cleaner inside an office, and we go in and set up the instrument, and we measure for the office background for a period of time. And then we turn on the device and we turn off the device. So during these measurements, nobody is present in the office, except that at the beginning, the researchers went into the office to set up all these instruments. And this here is a large set of analytical instruments that we use to characterize the both the gas phase and the particle phase compounds. So for the gas phase species, we have the ozone monitor, we have a NO2 NOx monitor, we also have a CAPS NO2 monitor, and the last one is a chemical ionization mass spectrometer, which is going to be measuring all these gas phase organics. Remember, when I mentioned about organics, we are not referring to one compound, we are referring to many, many different compounds. So this instrument called the SIMS is capable of measuring different types of gas phase organics. And for the particle phase, we also have several different instruments. So first of all, we have this instrument called the Scanning Mobility Particle Sizer, which is called SMPS. And SMPS allow us to measure PM1 number and volume concentration. So remember, PM1 is particles with diameter smaller than 1 micron. We also have a CPC, which is the condensation particle counter, which can measure total particle number concentrations. And by total, this instrument is capable of measuring particles up to around 3 micron in diameter. And last but not least, we have the aerosol mass spectrometer, AMS, that allow us to measure the chemical composition of these submicron particles. And again, remember, the aerosols are not made the same. They all have very different chemical components. So this AMS will allow us to gain some understanding of what type of aerosols are being made, if any of them are being made. So one note is that all of these instruments are real-time instruments. That means they're continuously taking samples and taking data from the office. So that's why we will be able to have continuous characterization of both the gas phase and particle phase composition. So just a few introductions about these mass spectrometers. So the first one is the SIMS, which is the chemical ionization mass spectrometer. And this instrument allows measurements of speciated gas and aerosol composition at the molecular level. So it tells you a lot of information about these organic species. And the gas phase data are being obtained while the aerosols are being collected on a filter. And then you can also thermally desorb the aerosols and then analyze by the instrument. So for this particular study, we are only using the gas phase measurement capability of this instrument. And the next is the aerosol mass spectrometer, which is the AMS. And you can see a schematic of the AMS here, in which the samples are continuously being pulled into the instrument, going through a time of flight region and then being analyzed by the mass spectrometer. And you can see a picture of the instrument also on the right. And this instrument provides real-time continuous quantitative measurement of these submicron non-refractory aerosols. And these are 5 different types. And you can see that organics and sulfate and nitrate, ammonium, and chloride. And because this is a high resolution instrument, we can also get the elemental composition of the aerosols. By elemental composition, I mean the hydrogen to carbon ratio, oxygen to carbon ratio. So if a compound or species has a high oxygen to carbon ratio, we'll say that it is very oxygenated. So this instrument will be able to give us that information. And so the bottom 2 figures shows you the type of information we get from this instrument. So because this is a mass spectrometer, so you get a mass spectrum, so you see different sticks, and the green are the organics, the red is the sulfate, the blue is the nitrate, and so on and so forth. And each stick represents one chemical formula or species. And this instrument is a time of flight instrument, so you can also get some sense about the size distribution of the different types of aerosols. So now let's take a look at what we found. We'll start with the gas phase compounds. So the mass spectrum that you're looking at is a mass spectrum measured by the SIMS, which is the gas phase species. And you can pretty much see a lot of different states at different mass to charge ratios. So the y-axis is pretty much the signal, the x-axis is the mass to charge ratio. So you can see that even in the office background, there's quite a rich spectrum in the office background, and there's presence of different chemical species in there. So now we go on and turn on this electronic air cleaner, and we want to characterize what happened to this gas phase species. So what we saw here is that there are some species that actually increase in signal. So they are higher and more abundant when we turn on this electronic air cleaner, including formic acid, which is these species here, and many of these are also labeled with their prospective chemical formula. However, at the same time, we also see that there are actually some gas phase compounds that actually decrease in signal during the device operation, such as these ones are labeled here, and we didn't label everything, but the major ones are put in the label here. So you can already notice that when we turn on the device, there's quite a lot of chemistry that is happening very quickly, that there's something that is being formed and something that is being decomposed. So let's take a look at the time series of these species more closely. So we're going to talk about 3 different classes of compounds, of 3 different classes of gas phase species. So the first one is this glyceraldehyde and glycerol. So for the rest of the talk, you're going to see this yellow bar quite a lot, and this is the notation for when the air cleaner was turned on. So the white part before the air was before the air cleaner was turned on. The yellow part was when the air cleaner was turned on, and the last white part was when the air cleaner was turned off. So you can see that there's this enhanced signal of this glyceraldehyde and glycerol at the beginning of the experiment. So these compounds are actually, a lot of them are the intermediates in metabolism, and also they have been widely used in cosmetics or additives in food. So what happened is that when the researchers went into the office to set up this experiment, probably these are coming from the researchers. And so you can see that the signal decreases, once they finish setting up the experiment and then left the office. So this is likely due to the presence of people in the office. And then we have the second class of compound that you can see that there's the immediate formation when the device was turned on. And lists are here a number of compounds. So specifically, I want to kind of just point your attentions to the formic acid and acetic acid as an example. So this lighter blue color and this darker blue color. So you can see that they are formed pretty immediately as soon as the device was turned on. And many of these species are all acidic species. You can see from their formulas here. So this little schematic here shows the structure of the formic acid, and this one is acetic acid. So you can see that they have this COOH functionality. So that is an acid functionality. And we also see the increase of HONO, which is this nitrous acid in this green color here. So these classes of compound are oxygenated products, and we call them oxidized VOCs or oxygenated products because if you look at their formula, there are quite a number of oxygen atoms. So, for example, formic acid has 2 oxygen atoms and 1 carbon. So for the O to C ratio is 2 to 1. So it's very oxygenated. And there's another class of compound that actually decrease during the device operation. So shown here are some examples of those compounds. So first of all, let's take a look at the hydrogen peroxide. Which is in the one in black here. So you might remember this device is supposed to generate hydroxy radical and hydrogen peroxide, but we actually observed that its concentration decreased during the operation. So it is possible that there is the reaction that is happening between the hydrogen peroxide and also the hydroxy radical. The hydrogen peroxide in the background of the office is reacting with the hydroxy radical given out by the device and resulting in a decrease in the hydrogen peroxide. And you can also see these big changes in the dinitrogen pentoxide. This is the darker gray color here, and also the lighter gray, which is the nitric acid, the HNO3, and also N2O5. So in the previous slide, you saw that HONO, nitrous acid, increases. And here you see this nitric acid and N2O5 decreases. And this could be due to surface chemistry. Some previous papers have reported that surface chemistry in the indoor environment can be a source of HONO and a sink for nitric acid and N2O5 that could explain some of the trends that we are observing here. So that's so much about the gas phase observations. So what about the aerosols, the particles? Do we see any changes when we turn on the device? So for the next couple of slides, we're going to focus on what happened to the aerosols in this office environment. So the figure here that you can see, the aerosol number concentrations is how many particles per cubic centimeter in that room. So the ones in the solid circles in red are the SMPS measurements. So remember the SMPS measure particles with diameter smaller than 1 micron. And the black line corresponds to the CPC measurements, corresponding to particles with diameter roughly smaller than 3 micron. And you can see that they are pretty similar. So what this means is that a lot of the particles in that room are mostly below 1 micron in diameter. And you can also see that the levels is about 500, 600 particles per cc. So it is fairly clean in terms of this particular office. And just to give you some context, how does this 500 particles per cc compare to different indoor environments? So recently we finished a study in collaboration with Delta Airlines to look at the different indoor aerosol concentrations. So we actually conducted many measurements in offices on Georgia Tech campus. So on the left panel, you can see that these are kind of the total particle number concentration in different offices on campus. And for this particular air cleaner study, our offices is the concentration is similar to these cleaner offices in these previous studies. And relatively speaking, the aerosol number concentration can really vary quite a lot depending on the different indoor environments. For example, this one is the measurements taken on board of Delta planes. And then there's also the measurement that we did in retail stores, grocery stores, and restaurants, and so on and so forth. And generally, just to recognize that the background aerosol concentration can vary quite a lot. And in this study, we did see that the in-flight aerosol is the lowest among all the measurements that we have done. So then what happened in this office that we are studying now when we turn on the electronic air cleaner? And now you can see this yellow region again. And you can see that as soon as the cleaner was turned on, there's rapid increase in the particle number concentration. And as soon as just turn off, the particle number concentration goes down again. So it increases from about 600 particles per cc to all the way about 4,000 particles per cc. And this is a rapid increase, but the increase is not as huge, maybe as in other environments that potentially when we use this particular electronic air cleaner. So now, what about the aerosol volume concentration? So now you see this blue color, which is the SMPS volume concentration. And you can also see that as soon as the instrument was turned on, there's this fast increase in the aerosol volume concentration. So it's not just that we're getting more particles, but the total volume of these particles also getting bigger when we turn on this device. So remember earlier I mentioned that we also had the mass spectrometers to kind of tell us what type of particles are being formed, if there's any, right? So before now we see that when the device was on, we got the increase in the total particle number concentration and the increase in the total volume concentration. So what are these particles? And this particular aerosol mass spectrometer will be able to provide that insight. So let's walk through this figure together. So the y-axis corresponds to the mass concentration of different types of aerosols, and the x-axis is just the time. So let's take a look at before we use the cleaner. So you can see that the background of the office is again pretty clean. It has about 0.5 microgram per cubic meter of organic aerosol here in green. And we have a little bit of inorganic aerosol, a little bit of sulfate in red here, and then barely any nitrate or ammonium or chloride. And in total, about 3/4 of the aerosols in the office background is organic aerosols. So what happened when we turn on the device? So if you now look at this green, yellow part, you can see that as soon as we turn on the device, there's quite a lot of increase in the different types of aerosol. So it's not just one type of aerosol that increased, but several different types of aerosols also show increase. So obviously, the biggest increase is this organic aerosol. So it increased from 0.5 microgram per cubic meter to about 2 microgram per cubic meter. And you can also see that there's a lot of little increase for the other types of aerosol. So the sulfate has a little increase. The nitrate here at the bottom shows a more obvious increase. The chloride in pink also increases. And ammonium kind of just pan out at the bottom, so barely show any increase. So we didn't really see a noticeable increase in ammonium. So taken together, all these aerosol composition measurements show us that there's this formation of secondary organic aerosols. Remember at the beginning of my introduction slide, the secondary organic aerosols are formed from complex chemistry in the atmosphere, or in this case, the complex chemistry that is taking place in this office. So let's take a little closer look at what kind of organics are we actually forming. So what you are seeing here is an aerosol mass spectrum measured by the aerosol mass spectrometer. So again, the y-axis is just the intensity of the signals, and the x-axis is the mass to charge ratio. And the gray sticks are the office background, and the green sticks, which is the skinnier green sticks, are the device operation. So as a first look, you can see that the fingerprints of the office background and the device operation are quite similar. So it's not like a major shift in the mass spectral features. But one big difference is that you can pay attention at this stick at mass to charge 44 here. So the gray is here, which is about 0.13, and the green is here all the way about 0.3. So what this shows is that there's this large enhancement in mass to charge 44. So in the mass spectrometer, where does the mass to charge 44 come from? So it comes from CO2 fragments. So CO2 is corresponding to 44. And in the instrument, the CO2 comes from the thermal decomposition of organic acids in the instrument. So organic acids have this functionality of COOH, and this breaks here, so that's why you get a CO2. So what this shows is that aerosols are really much more oxidized during the device operations. And you might be wondering what is this particular stick? So this stick is 28 CO, and in the data analysis, we set the signal of the 28 to be 44. So that is just part of the data analysis procedure, and that's why this MC28, or this first stick, is also the same height as the second stick. So remember, this is a high-resolution instrument, so we can actually go and calculate the elemental compositions and the elemental ratios. And shown here are two different types of elemental ratios, the O to C and H to C. So pretty much in the previous slide, you see each stick, right? So 44 is CO2. We can calculate the O to C ratio, which will be 2, right? And then for each stick, we can calculate this ratio, and we can sum them up and kind of get a sense of the total elemental ratio of the aerosols in that room. And you can see that as soon as the device was turned on, we observed this increase in the O to C ratio in this brown color here, and the decrease of this H2C ratio. So what this signifies is that as soon as the device turned on, the aerosols in that room become much, much more oxidized, that you see the sharp increase in the O to C ratio. So there's a way to kind of visualize this in a more, I'll say, coherent, summative way. So we use this diagram called the Van Craven diagram to kind of indicate these chemical transformations of the aerosols. So we'll walk through this a little bit together. So the Van Craven diagram, you can see the y-axis is the H2C ratio, and the x-axis is the O to C ratio. And you can see these are three horizontal lines here, one here, and then there's another line here with a slope of -1, another line here with a slope of -2. So what happened is that for the chemical transformations in the Van Craven diagram, depending on what chemical transformation is happening, they can follow these prescribed slopes. So for example, if your data, you put all your data on this figure and you kind of follow this -1 slope, you can indicate that you are increasing in carboxylic acid. And remember, carboxylic acid is COOH, right? So it could also signify the simultaneous addition of CO or NCONOH. So basically, the use of this Van Craven diagram allows us to kind of visualize the chemical transformation better. And you can also see this dotted lines, which is called the OSC, and this is the carbon oxidation state. And these dotted lines are basically calculated from the values of the H to C ratio and the O to C ratio. And the higher the oxidation state, again, means that the aerosols is much more oxidized. So before we look at the office data, let's look at where the ambient data all fall. So you can see this green triangle space. So this green triangle space is where a lot of the ambient data around the world, if you put all the data here, they fall into this little wedge of the green triangle. So what about the data from the office? So these are the data from the office. So you see the blue dotted points. So these are the data in the office background. So it's already kind of fairly oxidized to start with. And these red data points here at the tip here are the data when the device is turned on. So if you put all this together, what happened is that you have an office background here, and then as you turn on the device, it kind of move along this direction, right? Which is along this black arrow direction with a slope of around -1. So remember, I mentioned with a slope of -1 of transformation, you're likely adding carboxylic acid or adding carbonate and hydroxyuric and OH alcohol functional group at the same time. But because we are seeing all these acids from our instruments, it is likely that we are making carboxylic acids in the particle also. And one thing to note is that if we look at these red data points, the O to C ratio is about 1.3. This is a very high ratio, and this is very oxygenated. And you can see that it almost exceeded the tip of the green triangle, which is the ambient data. So these aerosols form in the office are some of the highest oxidized aerosol that we have ever observed by our group and other groups in the community. So what is actually happening then? So when we think of VOC oxidations, so we did not measure VOC in this particular study, but there are many studies measuring VOCs in different environments, including offices. And there can be different types of VOCs in the offices, such as silenes and toluenes. So what happened when a VOC encounter an oxidant? Does it really just go to carbon dioxide and water? So theoretically, the carbon in the VOC can be oxidized to become carbon dioxide. However, this does not happen too quickly and too easily. So this is more of what happens when a VOC is oxidized. This is the complex peroxy radical chemistry. So you got this VOC starting here at the top left-hand corner. So when a VOC is oxidized, you form an LQ radical, which is this R dot. And because we live in an environment full of oxygen, so this R dot very quickly grabs an oxygen and makes an RO2, which is a peroxy radical here in the center circled. And now this peroxy radical RO2 has many different options to continue the reactions. It can react with HO2 in green here. It can react with itself or react with other types of RO2 if there are other VOCs around. And if you're in a polluted places or in a room that has a lot of nitrogen oxides, the RO2 can also react with NO and NO2 here at the bottom. Or the RO2 can undergo this unimolecular isomerization by itself. So you can see there's this rich complex chemistry that can really take place. So you started with one VOC, and very quickly you end up with peroxides, carboxylic acids, organic nitrates, alcohols, carbonyls, and so on and so forth. And this is the complex multi-step VOC oxidation chemistry. And here I just give you an example of toluene. So toluene is a common VOC that is found in office environments and many other indoor environments. So the red box here is toluene. And what happened is that you can actually have an OH radical reactions, and then it will make all these different products very quickly. So you have one compound, and then you have hundreds and thousands of compounds. And the schematic here is by no means the full mechanism. If I were to draw the full mechanism, it would take up probably a whole wall at least. So this is just a really subset of the mechanism, just to give you an idea of what kind of products can be formed. And many of these are oxygenated VOCs. As you can see, a lot of oxygen atoms hanging out everywhere. And many of these will also have low volatilities. And so they don't want to continue to stay in the gas phase. And at some point, they want to be an aerosol. And that's kind of also where the SOA is being formed. So this is more of a realistic cartoon of what is happening. When a VOC in the office is being oxidized by the hydroxy radical generated by this electronic air cleaner, we are observing a large suite of compounds that is being formed. You have A, B, C, D, E, F, G, and keep going on and so forth. And a lot of these compounds that we observe using our instrumentation are these carboxylic acids. So these oxidized VOCs. And many of them have very low volatility, so they want to become a particle. And the office space that we measure is fairly clean. You might remember it's a pretty clean environment to start with. So there are not many other particles for these oxidized VOCs to condense on. And so that's why we're observing nucleation in which new particles are being formed. And once these particles are being formed, more gases will condense on the particle. And so these particles will continue to grow. So what we are seeing is really these contribution of carboxylic acids that are being formed from the chemistry and then making new particles and SOA in this particular environment. So other than the organic aerosols, remember we also see the increases in in nitrate, in sulfate, in chloride, but we did not see an increase in ammonia. So from the aerosol mass spectrometer point of view, the nitrate can fragment to two different states, NO and NO2. And by observing the ratio of this NO, NO2, we can tell whether it's an organic nitrate or inorganic nitrate. By inorganic nitrate, I mean the nitrate is associated with ammonia. So it's ammonium nitrate. So are we seeing organic nitrate or are we seeing ammonium nitrate? And the figure here shows you the ratio of the NO, NO2. It's a little bit noisy, But you can see this is what was the signal before the device was turned on. And now when the device is turned on, you can see this increase in NO2 ratio. And this ratio is on average about 17. It is very high. So based on our measurements before and all the knowledge in the atmospheric chemistry, this high NO2 signified the presence of these organic nitrates. So it's interesting that we're seeing even organic nitrate formation in an indoor environment. Organic nitrate is fairly prevalent in the outdoor environment, and it is pretty ubiquitous in the ambient. And what about sulfate? So previously, we also see that there is the contribution of organic sulfate, and inorganic sulfate can be teased out by the mass spectrometers. So in this particular triangle figure here, if you're getting closer to the origin, that means you are getting more and more organic sulfate. And again, by inorganic sulfate, I mean ammonium sulfate. So these are the data from the office. So the blue data points are the ones in the office background, and the red data points are the ones when we turn on the device. So there's this indication that the data points are getting closer to the origin as the device was turned on, and that these two fragments decreases, and that's why it makes the data move towards the origin. And based on this analysis we have done before, this suggests that we are also forming organic sulfate in this office environment when the device was in operation. So many of these secondary species, either in the gas phase or particle phase, can have adverse health effects. In terms of the health effects of SOA, so SOA has been reported to induce cellular reactive oxygen species generation, cytokine production, and the oxidative modification of RNA, and so on and so forth. And in the figures here, I showed two figures from some of our previous work. So here in the left, you can see a figure showing the different cytokine productions, the TNF-alpha and IL-6, and the different colors just means different type of aerosols, different type of SOA. And you can see that they induce a diverse set of cytokine response depending on what type of SOA you are and what kind of reaction conditions that these SOA are being formed. And in the figure on the right here, you see the y-axis is the ROS, which is ROS and RNS, which is the reactive oxygen species and reactive nitrogen species generation. And the x-axis is the carbon oxidation state. Remember those slashed dotted lines in the Van Craven diagram, and that the more oxidized you are, the higher the carbon oxidation state is. And what we found in this previous work is that the aerosol toxicity increases with carbon oxidation state following this line for this type of aerosol. And just a reminder, the carbon oxidation state of aerosol formed in offices is after the device is turned on is about here. So the oxidation state is really high, and so it is really important to understand, what are some of these potential health effects of being exposed to these secondary byproducts that are being formed in this particular environment. So just to conclude, to our knowledge, this is the first study that monitor the composition of both the secondary gas phase products in both the gas phase and particle phase during the operation of an electronic air cleaner in a real-world setting. And this particular electronic air cleaner that we tested is a hydroxyl radical generator. And what we observe here is that there's a rich chemistry as soon as we turn on this air cleaner. There's the rapid formation of all these oxidized VOCs, especially these low molecular weight organic acids. We also observe a rapid increase in the particle number and mass concentrations, and which corresponds to the formation of these secondary organic aerosol, SOA, that is formed from these complex oxidation chemistry. And as I mentioned, we also observe the formation of organic nitrate, organic sulfate, and chloride. And this device that we tested is actually quite similar to many other commercially available devices, and so similar experiments should definitely be conducted with other devices. And just a few more thoughts. So hopefully today you see that the VOC oxidation is not as simple as changing everything to carbon dioxide and water in one step. In reality, the VOC oxidation proceeds via complex and these multi-step chemistry, and they're forming a whole host of oxidized VOCs along the way, which can have low volatility and end up in the formation of these secondary organic aerosol particles. And so there is really the potential for electronic air cleaners to induce complex indoor chemistry. And in the process of using these, unintentionally forming more pollutants in the indoor environment. So when we discuss electronic air cleaners and chemistry and chemical compositions or unwanted chemistry, a lot of the time the focus is on ozone. But it is important to note that ozone is not the only chemical of concern. And you can see in today's presentation, there are many other chemicals that can actually be formed other than ozone. And so if a unit does not produce ozone, which is good, but it does not mean that the unit does not produce any other chemical of concern. And many of these secondary species are reported to have detrimental health effects. And so future studies on air cleaning technologies should not be limited to testing whether the unit can inactivate bioaerosols or reduce VOCs. So these are important. However, at the same time, we should also test whether these units can actually generate these oxidized VOCs and secondary organic aerosols in different indoor environments. And the hope is that we can actually eventually maybe establish some sort of testing protocols and standards so there can be more guidelines and regulations for everyone to choose the proper type of air cleaning technologies. And with this, more peer review studies are really needed by different communities. And in the meantime, I would recommend just sticking with mechanical filtration to remove particles. And HEPA filters are very efficient in removing particles in different environments. And this work has been published in Environmental Science and Technology Letters recently. You can click on this link to read the full paper, and the paper has been made openly and freely accessible through the ACS COVID-19 subset. And there's also a Georgia Tech Press article that goes with it if you're interested. And lastly, I would like to thank Georgia Tech COVID-19 Rapid Response Fund for supporting this important work. And the chemical ionization mass spectrometer in which we use to characterize many of these oxidized VOCs in the gas phase was purchased through an NSF major research instrumentation grant. And I would like to thank my colleagues, Jen Kaiser and her groups for helpful discussions on the experimental setup, and for Jay Lee for help with this nice illustration of the office setup. And this work is led by a group of really talented and dedicated students and postdocs. And the work lead author is Taekyu Ju, who's a graduate student in Earth and Atmospheric Science. And then the contributing authors are Jean here, and also Daniel here, and Sabrina here. And with this, I would like to thank your attention, and I would take questions.
Thank you so much, Sally. That was an excellent presentation, and we've gotten a lot of questions, definitely a lot of interest from the audience. So I will jump right into those. First off, we had a few questions. I think you spoke a little bit about this at the end, but we had quite a few questions about how the results of this study might compare to other electronic air cleaners, like ionizers or plasma-based devices, some of these other technologies that are out there. Do you have a sense of how those other devices might compare?
Yep. So let me go back and find my slide on some of these earlier studies. So for this particular slide, if you can see my screen, I cited two prior work on the potential secondary products that is generated from these other electronic air cleaners. So in this particular one is a bipolar ionization device, and this one is an ion generator. So in this two work, so first of here, the authors, they characterize these oxidized VOCs and also VOCs. And as you can see from this figure, the downstream, the bar, is already quite different from the upstream bar. So there is definitely some chemistry happening here. And they did not characterize the particle composition, but I would imagine there could potentially be also some particle formation. And in this particular study, the researchers also generated these figures showing that the ozone is increasing, the oxidized VOC, so this one is the formaldehyde and this one is the non-NL. It also increases during the operation of these electronic air cleaners. So I would say that while the studies in general are still pretty limited, many of these studies have indicated there is all this potential chemistry that is happening. And in terms of really characterizing the composition of these gases and the composition of the aerosols, not just the total number concentration, but the composition of the aerosols requires these the use of these mass spectrometers that we reported here. So hopefully this will allow for more researchers to try to do more of these studies to really thoroughly calibrate or characterize these different types of electronic air cleaners before we can widely adopt these technologies in our everyday use.
Great. Yeah, definitely. There's definitely more to be done in this area. So thank you. There's a couple of questions here related to ventilation. So we know that air cleaners are often used in combination with ventilation and other strategies to help reduce the transmission of the virus that causes COVID-19. So we had some questions, was the room that you tested in ventilated with outdoor air, and kind of how might that change the results that you saw here if there's higher outdoor ventilation rates, given that there's some of these compounds are also found in outdoor air?
Yep, exactly. Yeah, so that is definitely important that you should have enough ventilation in an environment, that these portable cleaners are supposed to just be a supplement. So in this particular office environment, we have active ventilation. And in terms of doing the experiment, we make sure that when we are doing the experiment, that there's no change in the ventilation. So before we turn on the air cleaner, during the air cleaner usage, and after the air cleaner, that the ventilation in the room is staying fairly constant. And obviously, as I mentioned also in my talk, there could be incursion of aerosols from outside, right? But as you can see in our data, let me just bring out that slide. As you can see here, right, that is rapid increase in the particle number concentrations as soon as the device is turned on. So this is probably not because of the ventilation change, because we keep the ventilation fairly constant during this period. And you can also see this rapid increase in particular organic aerosols. So I think in order to do these tests, that's why I mentioned that we need to have more standards and guidelines in terms of standardizing how to do these tests. And so in this particular work, we did our best to make sure that the environment is stable during this experiment, so we can actually really characterize the change, if any, from the use of these cleaners.
Great. Let's see. There was another question kind of about the testing that you did itself. The time that you operated the air cleaner seemed to be about 2.5 hours, and one of our listeners was interested in kind of how you determined that time, and if you would expect the byproducts to kind of continue to generate or level off or something if the device was left on longer, as it might be in the real world.
Yeah, so we just tested for a couple hours. Is just like the availability of instruments and all that stuff. But you can see that we kind of, our hypothesis going into this experiment is to evaluate if there's any byproducts being formed in the gas phase and in particle phase. And from the data that we have already is very clear that there are gas phase and particle phase byproduct that is being formed. As you can see in this time series, that the organic aerosol and the particle number and the gas phase is start very rapidly increasing. And then it kind of just maybe the slope decreases a little bit. And if we let this running on for longer time, it might just reach a plateau. And remember how much of these byproducts are formed depending on how much raw materials you have in the office, so to speak, right? So in this case, we are talking about the oxidation of the VOCs in the room by the hydroxyl radical. So this office is actually pretty clean to start with. So there's probably not that much VOC in the office background for these hydroxyl radical to oxidize. And you can see that there's this increase in organics and particle number concentration, but the increase is not huge. I mean, at the end, it's still 2 microgram per cubic meter. But one thing is that if we deploy these electronic air cleaners in an environment with a large amount of VOCs, maybe in schools or in restaurants and other indoor environments with a lot of people, because remember, in this experiment, we also did not have anyone in the office, then there's a lot of raw materials, VOCs, for the hydroxyl radical to react with. And in that case, I would expect much bigger increase in both the particle number concentration and the gas phase species. So this really kind of coming back to, when we are deploying these different units in different environments, the chemistry can be very different depending on what kind of environment we have to start with.
Yeah, definitely. Yeah, there's definitely, this was a pretty clean room, so probably some different scenarios out there. Another question we had was, how does the oxidative chemistry that you saw during the operation of this device compare to the chemistry that's driven by ozone?
Yeah, so the chemistry that we are looking at is the hydroxyl radical chemistry, right? But the other two types of very important oxidants are ozone and nitrate radical. So in an indoor environment, we can also have ozone. So when ozone reacts with an VOC, it can also induce a very series of chemical reactions that is complex. That's one of the slides that I showed. And the ozone oxidation chemistry with VOCs can also induce these compounds that have low volatility and that would eventually forming SOA. So we routinely see this in the ambient environment, see this in our lab, that the ozone chemistry can definitely induce very complex VOC oxidation chemistries forming these secondary products.
Great, thank you. A couple of questions came in about kind of how, some air cleaners combine different technologies. So maybe have a hydroxyl generator, and maybe there's some other technology like a activated carbon filter or mechanical filter. Do you think the results would be any different if you had a technology that combined some of these other filter types with this, something like a hydroxyl generator that you tested?
Yeah, so the question is, how do we contain that? So for this particular device, so let me see if I can pull out the cartoon showing how it looked like. So if you see this little fan here, right? So the unit is kind of dissipating the hydroxyl radical into the room. So with that, the chemistry is happening in the room. So in that case, then it's going to be hard to contain those secondary products. But if there is some sort of device that would allow the potentially secondary products to be scrubbed before they get emitted into the room, that might work. But in this particular situation, if you have a device that dissipates chemicals into a space, that'll be very hard to contain on these secondary products.
So there was a couple questions about the health effects that you talked about at the end. Ozone is not the only pollutant that we are concerned about here. So a couple of listeners asked, what's kind of another, high concern chemical that you might have seen here, or what other types of experiments might be done to investigate that a little further?
Yeah, so I would say that ozone is definitely, not the only chemical of concern. And if a unit does not emit ozone, that is great to start with, but that does not mean that, we don't pay attention to other chemical species. And of course, PM 2.5 is regulated by EPA, so the particles themselves are not good for you to breathe in. And the fact that you have some particles in the ambient environment does not mean that you want to make more of them unintentionally. So the PM particles and SOA are definitely another chemicals of concern. And as I showed that in one of these slides, that there are some studies, including some from our group in literature, showing the potential health impacts of these secondary organic aerosols. And of course, the gases are also of concern. You can see in this talk, there are many oxidized VOCs that have been generated because of the chemistry that is induced by these hydroxyl radical generator device. So these species have also been shown in previous studies to have adverse health effects. So that's why I think there's really a need for a sustained, more comprehensive effort to really characterize the chemical reactions that can be induced by different type of electronic air cleaners and carefully evaluate their potential risk for being exposed to these secondary products, either in the gas phase or particles that is being generated. And I think at the end, there needs to be a more, as a holistic view of kind of looking at all these different air cleaning technologies. It is good that we are deploying additional air cleaning technologies to ensure a healthy indoor environment. And of course, some of these devices could potentially also inactivate the virus, which is a good thing. But at the same time, if they're inducing secondary chemistry and making all these secondary gases or particles that have also health effects, then we need to kind of find a way to comprehensively evaluate as a big picture of the pros and cons of these different devices. But in the meantime, I really suggest stick with the HEPA filters. They are very good at filtering all particles of all sizes, and that they did not involve any chemistry in terms of their operation. So that would be my recommendation at the moment.
Okay, definitely. We also had a couple of questions about how humidity might affect the results of the study. If you kind of have a sense of what the humidity was in the room during the test, how that might impact the results. And I guess these are sometimes recommended to be used with humidity generators. So.
Yep, exactly. So I can't speak in terms of the humidity effect of these air cleaners in terms of inactivating our virus. So that is not what we study, but humidity can definitely play a role in these oxidation chemistry that I'm talking about. So we didn't measure the humidity in that room, but we are in Atlanta, it's summer, so it's actually in spring. So I would say it's probably like 40% humidity, roughly speaking, if I were to guess. But humidity definitely plays a role in the VOC oxidation chemistry. And that's what we know from all the studies we have done by our group and other groups in terms of the hydroxy radical reacting with VOCs. This is actually a good slide. So if you see the slide here, you got the dry and humid and dry, right? So actually these are aerosols formed under different humid conditions. And you can see that they have different cytokines being generated. So how the humidity can affect this oxidation chemistry can be manifolds. So first of all, the gas phase reactions, let me bring out this point, right? When a VOC reacts with an oxidant, whether it's a hydroxy radical, ozone or nitrate radical, the gas phase chemistry can be affected by the presence of water vapor. And once you form an aerosol, if your aerosol actually has particle water, you can also have aqueous chemistry happening in the particle phase. So the composition of your gas phase species and the particle phase compounds can be very different depending on whether you have high humidity environment or not. So this would definitely change these chemical compositions. And if the composition is changed, the products are different, and they could potentially also have different health impacts, as you can see from this last slide, in which the cytokines measured by these different type of aerosols under different humidities are very different. So that is definitely, I would say, another parameter to definitely consider.
Great. We had one more question. It's kind of going back to the question we had about ventilation. Some folks are using air cleaners in low ventilation environments to kind of bring up the equivalent air changes per hour in those environments, and using carbon dioxide as an indicator of doing this successfully. Did you see any change in carbon dioxide in the operation of this device?
Yeah, so we did not measure the carbon dioxide in this particular study, but at Georgia Tech right now, we actually have deployed air cleaners in every classroom because semester just started several weeks ago, and we have a full in-person teaching component. And so we actually install these portable air cleaners with HEPA filters in each of the classroom. And we also have CO2 sensors and also these low-cost aerosol sensors to measure the air exchange rate. So with the use of the portable filters, like say for HEPA filter, so HEPA filter is going to increase the decay of the aerosols. So with the use of a HEPA filter, portable filter, you're going to see the aerosol decays faster when the people or students leave the classroom. So for the CO2, it's a combination, obviously, of many factors, but the filter itself is not going to change your CO2 concentration. It's just really, in order to reduce the CO2 concentration, it is really important to bring in as much fresh outdoor air as possible. So in the ventilation system of the building, if there is a way for you to increase the relative intake of outdoor air, so that should be increased. Or if you can open the window and not being affecting other rooms in that building, then it's also advised that you should have the windows opened. But I would say that definitely if you have very limited ventilation in the buildings or schools that you have, it is really important to bring in these HEPA filters to try to increase that turnover rate of the aerosols, that you see faster decay of these aerosols.
Great. So another question about the different types of technologies that are available. Another thing that's used is germicidal UVC. Do you have a sense for whether that might also generate some of these pollutants?
Yeah, so that one, I don't recall from the top of my head if they actually can potentially generate, induce these chemistries. So one thing is also kind of pay attention to the wavelength of the UV that is being used, because a lot of these chemistry can be induced by smaller wavelengths. For example, like the if your UV is in the UVC region, which is like around 300 nanometers, that can actually induce some chemistry in these environments. So I think the type of UV is also important, but I can't recall from the top of my head if there are studies specifically regarding the potential secondary chemistries from these devices.
Okay. I have another question here. We talked a bit about the environment being relatively clean in this case, and one of the kind of concurrent events that we're having going on along with the COVID-19 pandemic is that we're having a lot of wildfire smoke, especially in the western states. How might the use of this type of device, this type of electronic air cleaner, change in that setting where there's a lot of wildfire smoke that might be getting indoors?
Yep. So I would say that can definitely compound the situation. So imagine if you are in a house that is close to these wildfire emissions, and that if your house is not very well sealed, you're going to get some incursion, from the outdoor air into your, living environment. And many of these wildfire emissions contains a whole host of VOCs. So they are the perfect raw ingredients to be reacted with these hydroxy radicals in your room if you have a source of hydroxy radical, and that would generate a lot of these secondary gases and aerosols. So what you're pretty much doing is that you're bringing the outdoor pollution indoors, so to speak, right? Because if you don't have a lot of oxidants in your room, in your indoor environment, then maybe these VOCs will just stay as VOCs. I mean, they're not good for you, but they will stay as VOCs. But if you have a source of hydroxy radical, then this chemistry that we are talking about today will definitely take place. And you're going to produce new particles, new PM, and a new secondary gas phase products. So then I would say that it kind of really worsens the indoor environment and the indoor air quality that you will be exposed to, certainly. And in that case, then having a HEPA filter would help.
Great. Let's see, we had another question. Going back to the toxicity experiments, just a couple of questions about those. Were they in vitro analyses, and if so, what types of cells were used or controls? If you could just talk a little bit more about that slide there.
Mm-hmm. So for these studies, we are using macrophage cells, and we have a 96 well plate. So we generate these aerosols. I'm not sure if you can see, but there's 6 different types of secondary organic aerosols that we generate in our control chamber in our lab. And basically, we are generating these SOAs under different environments, and we collect them on a filter, we extract them in the cell media, and we expose them to the macrophage cells. And we are measuring the cellular ROS that is being generated after exposure. We are also measuring the cytokine production after exposure. So that is, pretty much where these studies are from. And I didn't show it here, but we also have done studies in which we take the samples from the outdoor environment and expose to the cells, and also measure these ROS and cytokines. But personally, my group, we haven't collected the indoor samples and exposed to the cells and see what would happen with cellular ROS or cytokine generation. And this office probably did not generate enough mass to be collected, but I would imagine if a similar device is deployed in other environments with a lot of VOCs and a lot of people, and also, as you mentioned, maybe environment with a lot of wildfire emissions that can go indoor, I would imagine it can potentially generate enough aerosol mass to induce some of these similar health endpoints that we are measuring here.
Thank you. There was another question here, which I think is probably common to a lot of different devices. In your diagram, you kind of have the little fan to represent the device that you tested, and it's kind of blowing air and sort of, does that kind of move dust around or move particles around in the environment? How does that kind of impact, where those particles and things are going?
Yep. Yeah. So this one is a dissipating fan. So it's kind of helping to disperse these hydroxy radicals into the environment. And one thing that I mentioned at the beginning of the introduction is that these with suspension of dust can definitely, be a source of indoor particles. But these particles, the dust tend to be a much bigger particle. So these instruments that we are measuring here are mostly PM1 instruments, which are PM1 here. So PM1 is particles smaller than 1 micron. So, and we are seeing these organic aerosols very clearly. So this really allows us to pinpoint that this is not the dust particles that we are getting from the floor because of the fan blowing, but this is indeed due to chemistry, just because of the size and the composition that we are measuring here.
Great, thank you. All right, I have kind of one other question. So there's several questions about specific products or specific technologies from the listeners, and I don't want to necessarily put you on the spot about that, but I was kind of curious, what in an ideal world, kind of what information would you want to have for consumers to, when they're looking at these different devices, what kind of information would you want them to know before choosing one to buy for their situation?
Yeah, so I mean, this is a challenging question, and as I mentioned towards the end of my presentation, I feel like there's the need for this more comprehensive, like holistic effort to really evaluate the different aspects of these different types of cleaners. First of all, there's different parameters. There's different types of cleaners already. There's different technologies, and kind of the matrix that we can think of is that, first of all, does it produce ozone, right? Because ozone is right now one of the very few criteria that a lot of the manufacturers would say we do not produce ozone, which is a good thing. But the question is that I think there's a systematic investigation of the formation of these gases, pollutants, and also the potential particle generation as we are seeing here. And obviously, there's if the product is claimed to be able to inactivate virus, I think there's need to be a thorough investigation to make sure that this is really effective under different environments, not just the perfect testing environment in the lab in terms of its efficacy in activating pathogens. So I do think there needs to be this comprehensive effort to evaluate the different types of air cleaners with different parameters, kind of to score each parameter and say this is good, this is bad. But as of now, there's really a limitation of the number of studies that have been conducted. In this particular work, we study one type of electronic air cleaners under one environment, and there are many other parameters out there. So I feel like it's not going to happen overnight in terms of us having this clear picture of what is these different parameters of different performance with different type of electronic air cleaners. And so with the lack of, that's why there's this call for more studies to kind of have a better understanding of these chemically active air cleaning technologies. And until then, I would say that we stick with what we know that it works and that does not dissipate the chemicals into your environment. And with that, a HEPA filter is probably what I would recommend if you have a ways to, and if you want to install a portable air cleaners in any environments, schools, homes, just to stick with the HEPA filter, which does not dissipate any chemicals into your environment. Will be probably your best moment.
Great. We might be getting to the end of our questions. And so I think we'll kind of wrap it up here. So thank you, Sally, for presenting today. This has been a fantastic presentation, and thank you for sticking around for this Q&A. These electronic air cleaners have been of a lot of interest during the pandemic, so I thank you so much for sharing your knowledge and your experience with us, and I thank you also to everyone who joined us online for submitting your questions. I'll remind everyone for announcements of future IAQ science webinars to learn more about indoor air quality. You can visit epa.gov/IAQ. So thank you, everyone, and please enjoy the rest of your day.
Thank you.